Hi everyone, I am Negin Malekian, a PhD student at TU Dagestan, and here is the team for the project. Today I aim to show you how with only few sequences we could identify targets of quinolone resistance, not only the known ones but also some new ones. Before I go through my presentation, I would like to draw your attention to the global threat of antibiotic resistance, in specific fluoroquinolone resistance. As you can see in this map, in just 10 years, the amount of fluoroquinolone resistance significantly changed in Europe, especially in Eastern European countries. The first question is, how do fluoroquinolones work? They target GLBIO and parS. GLBIO and parS are enzymes which are responsible for introducing negative supercoils into DNA, a necessary step in DNA replication. This process involves cutting the double-stranded piece of DNA, passing another one through it, and then resealing the broken strand. Coinolones interact with this enzyme and prevent the resealing of DNA. This leads to the inhibition of DNA synthesis and eventually death of the cell. However, bacteria can be resistant to quinolone through the mutations shown in this figure. It is thought that this mutation alters the sites at which quinolone interact, thus reducing the affinity of the drug for the enzyme. However, it should be noted that the mechanism that we mentioned were not easy to understand. It took around 30 years from the discovery of the first quinolone to identifying its target. The first quinolone, which is nalidixic acid, discovered in 1962 by chance during the synthesis of an anti-malaria drug. 2 years later, GLBIO was discovered as the primary target of quinolones. In 1980, fluoroquinolone introduced to the market. Fluoroquinolones contain a fluorine in their chemical structure and are effective against both gram-negative and gram-positive bacteria. In 1990s, parS discovered as the secondary target for quinolones. So, as you can see, from the discovery of the nalidixic acid to the discovery of parS, it took 30 years, which is really a lot. The known mechanism of quinolone resistance in GLBIO and parS discovered with a hypothesis-driven approach. Although this is a good approach as the mechanism of resistance is well understood, it is limited only to the hypothesis. It is slow and it needs lots of knowledge. So, what is the alternative? The alternative is a hypothesis-free approach, which is comprehensive, fast, needs only few sequences, and it is systematic. However, it has also its own disadvantages, which is the mechanism of action is unknown and needs further study. Genome-wide association study is a systematic way to find the targets. Here you can see a simplified overview of the GBAS analysis. To conduct a GBAS analysis, we need some genomes and also their phenotype. By associating the genotypes with the phenotype using regression, we can identify the most significant variants. To be more exact, we need also to model the effects of population structure to make sure that the significant mutations reflecting association with the phenotype rather than association with the population subgroups. Here, I would like to introduce our dataset. Our dataset consists of 99 E. coli samples, which are collected from a Dagestan wastewater treatment plant in Germany, with resistant data against 4 quinolones. Here you can see the results of you can see the visualization of our results using Manhattan plot. The x-axis shows the chromosomal position, and the y-axis shows the minus like 10 of p-value for the variants, and the horizontal lines show the Bonferroni corrected p-value. As you can see, very few variants pass this threshold, 13 out of around 400,000 variants for all coinolones, reflecting that only a small portion of the genome contributes to the resistance phenotype. Now, the question is, what are these 13 variants? As you can see, the top ones with very significant p-value are the known variants in gyrase and parsi. After that, we have some new variants in bdcA and valS, etc. Here, we separated the synonymous from non-synonymous one, as it is easier to find a causality link for them. However, also non-synonymous can indirectly affect the phenotype. Also, you can see that we found less variants for nalidixic acid and ciprofloxacin, but it should be noted that all of these 13 significant variants had a very good p-value and high ranking for all the coinolones, but possibly because the p-value cutoff was very stringent, they did not appear for the other coinolones. This means that providing with more data, they possibly also pass the threshold for the other two coinolones. Now we need to look to the biological function of these variants. The first variant, non-synonymous one, was in bdcA. Also, bdcA so far is not linked directly to the antibiotic resistance. It can be indirectly related to antibiotic resistance. This is because bdcA is involved in biofilm dispersal, and biofilm formation is known to play a role in biofilm formation is known to play a role in developing antibiotic resistance. To shed more light on the possible causality of the bdcA variant, we look into its protein structure. Our interesting variant in bdcA is in the vicinity of the active site residues that you can see in the figure. Serine is bigger than glycine, and it may influence the loop there, and through that regulate the active site and disrupt the biofilm dispersal. We found also some synonymous variant in the valS gene. valS is a kind of ARS or tRNA ligase, and ARS is an enzyme that attaches the appropriate amino acid onto its corresponding tRNA. As you can see here, there is evidence that mutation in ARS enzymes increase ciprofloxacin resistance. This is happening by upregulating the expression of efflux pumps. Also, ARSs known to be promising drug targets for antibiotics. First, because they are highly conserved among bacteria. Second, they are evolutionarily divergent between prokaryotes and eukaryotes, meaning that we can develop antibiotics that specifically target bacteria, and also they have some good features in terms of drug development, like solubility, stability, and easiness of purifying. To summarize, we showed that although it took 30 years to find targets of quinolone resistance, we could find a target of quinolone resistance faster, cheaper, and using only few genomes by a high throughput hypothesis-free GWAS. Not only find we the known variants in gyrase and parsi, but also we find 10 new variants. A variant in bdcA suggests a novel resistance mechanism via biofilm dispersal. Also, we started to do experimental validation with a team from hydrobiology. Before I end my presentation, I would like to draw your attention to some more questions that we did not discuss now, but if you are interested, we can discuss it offline or you can read more about it in our paper on bioarchive. So the questions are, is bdcA gene and its variant is something limited only to our dataset from the SN or can we also observe it in other bacteria? Also, as bioinformation is a general approach, did we also observe the variant in bdcA correlate to other antibiotics other than Coinolones? How was the population structure model? How do pan and core genome compare to pan and core variome? And so on. Thank you.